So hello guys, welcome back to my YouTube channel, that is Learning Biophysics with Team 320. And today's my topic is it's a week 8, and today's my topic is mitochondria, structure and function. So when we start, these are the objectives which you see on the screen. So mitochondria are intercellular organelles found in almost all human cells. Mitochondria are thought to be derived from anaerobic bacteria that is invaded the proto-eukaryotic cell more than billions years ago and lived in a symbiotic relationship with them, exchanging energy in the form of adenosine triphosphate or ATP, we can say, for residence. However, this endosymbiotic hypothesis is not universally accepted and has been challenged. Each human cells contains an average 100 to 1000 of mitochondria. The exception is mature red blood cell, which really uh, contain no mitochondria. Although mitochondria were originally represented as individual isolated organelles, it is now recognized that mitochondria from a dynamic connective network, also known as reticulum or a syncytium. As zygote mitochondria are derived from the ovum, their inheritance is maternal. So when we see the structure of mitochondria, we have some compartments. So these are four main compartments: outer membrane that is permeable to certain ions and small molecules, intermembrane space that is composition similar to cytosol, inner membrane respiratory chain proteins are present. So inner membrane are folded into multiple crystals, allowing a large surface area, and the lastly that is matrix, where the most of metabolic reactions take place. That mitochondria DNA is found within DNA protein complexes called nucleoids. So in the mitochondria, now we will discuss about their functions. So mitochondria are intercellular organelles that are essential for aerobic metabolism and energy production. That's why we say it as a powerhouse of the cell, and it is also functions in oxidative phosphorylation, involves several metabolic pathways, that is beta oxidation, Krebs cycle, synthesis of iron sulfur uh, clusters, and maintains, replicate and transcribe their own DNA and translate messenger RNA or mRNA into protein, the import protein. So since most proteins that the mitochondria require are encoded by nuclear DNA and translated in the cytosol, so specifically human mitochondrial DNA is a double-stranded circular DNA containing approximately 16,500 base pairs. Compared with over billion nuclear DNA, it encodes for 13 protein subunits that are associated with the nuclear genes, to four enzyme complexes plus two ribosomal and 22 transfer RNAs that are needed for protein production by the intramitochondrial ribosomes. The ongoing remodeling of the mitochondrial network is also a function of a mitochondria. So we see additional roles, that is apoptosis, production of reactive oxygen species, calcium homeostasis, maintenance of lipid membrane, immunity. Mitochondria have an ineffective DNA repair system, and the mutation rate for the mitochondrial DNA is over 10 times the rate for nuclear DNA. Here are some mitochondrial markers, that are inner membrane synthetase and succinate dehydrogenase, ATP synthase, succinate and dehydrogenase, succinate. And inner membrane space have creatine kinase, adenyl kinase, and matrix makers are glutamate dehydrogenase, pyruvate dehydrogenase, enzyme of TCA cycle, and oxidation of fatty acids. So how proteins are imported into mitochondrial? There are two ways. One is translocators of inner membrane and outer membrane. Those import receptors are located. Protein passes through TOM and enters intermembrane space, then passes through TIM to enter the matrix. You can see on the video, it's how like protein is passing through inner membrane to outer membrane to the matrix, and peptidase how it binds with the protein and work with. This is the video. The TOM and the TIM alignment is also an important topic research suggests. Like outer membrane and diffuses laterally until it comes contact with the TOM or the pre-sequence complex. Protein can enter the matrix in an energy-dependent manner. The pre-sequence is clipped by the peptidase and chaperones assisted in folding proteins. Transmembrane domain can diffuse laterally out of TIM and embed in the inner membranes. So now you will think how it link with clinically. So we will see neurodegenerative disorders that is often caused by defects in translocases. Genetic disorder in TIM production result in the deafness and dystonia. Now let's discuss symbiogenesis. DNA and our ribosomes of mitochondria and prokaryotes have many similarities resulted in endosymbiotic theory of mitochondrial evolution. Now, mitochondria were originally independent prokaryotic bacteria. Here we can see that mitochondria can do this by oxidative phosphorylation. As a result, prokaryotic cell lost part of their DNA and ability to live independently. Eukaryotic host cell becomes dependent on energy produced by incorporated bacteria. Now we will see mitochondrial myopathies, that is characterized by an impaired energy production in form of ATP. Mainly affects organ with high energy demand, that is brain. These are extremely rare and prevalence, that is 13 is to 100,000. One of these myopathy is etiology caused by mutation defects in the mitochondrial DNA which are maternally inherited. Children of an affected mother will likewise be affected. Genetic expression is variable due to heteropathy. Heterogeneity of the mitochondrial DNA within a cell. It is caused by a selective distribution of mitochondria with mutated DNA and it result in variable expression of diseases that follow mitochondrial inheritance. Next myopathy is pathophysiology, that is mutation defects in mitochondrial DNA that leads to impaired oxidative phosphorylation, decreased production of ATP, and which further creates increased glycolysis and excessive production of pyruvate and accumulation of lactate and alanine occur. Organs with a high energy requirement are particularly affected, that is retina, brain, inner ear, skeletal muscle, and cardiac muscle. Some common clinical features that we can see that external ophthalmologia and some diagnostics are genetic studies, mitochondrial DNA, muscle biopsy, immunohistochemistry shows the red ragged fibers caused by the intermyofibril accumulation of defective mitochondria in muscles. Now we will see laboratory studies of these mitochondrial myopathies, that is normal creatine kinase, that is CK, elevated locate and alanine in serum, urine and or in CSF. Now we will discuss subtypes of mitochondrial myopathies, that is MELAS, that is MELAS, characterized by mitochondrial encephalomyopathy, lactic acidosis, recurring stroke-like episodes. Other findings include muscle weakness and tonic. Next, we can have that MERRF, characterized by myoclonic epilepsy and direct red fiber. Causes of this is point mutation of 8344 base pair of mitochondrial DNA. Specifically, in we can see in mitochondrial MTTK gene, that is destruction of import proteins involved in sedative phosphorylation. Now we will see seizures, generalized seizures. Now the final subtype is CPEO, characterized by chronic progressive external ophthalmoplegia with bilateral ptosis. Kern-Seren syndrome, that is characterized by ophthalmology and retinitis pigmentation, impaired electrical activity of heart, especially AV block, LHON, that is Leber hereditary optic. Now, epidemiology, that is most commonly occurs in teenagers and young adults. Male, if we see the male-female ratio, that is 3:1, and the cause of this is cellular death in the optic nerve neurons. Clinical features is painless, acute or subacute bilateral visionless. Next syndrome is the Lee syndrome. That is clinical feature we can see vomiting, diarrhea, dysphagia or failure to thrive, hypotonia, dystonia, and the ataxia. And we also can see the rapidly progression psychomotor regression and ophthalm and is that optic atrophy, that is the we can see. Death usually occurs in 2-3 years to the respiratory failure. So that's the end of my topic. So I hope you all have liked this video. So please do share and subscribe. Thank you.